What I'd like to do with my presentation is to go ahead and really use Nick's presentation, Amita's presentation, John and Jeff's presentations to really go a little bit deeper and talk a little bit more about PSTC and really the power of collaboration, what we've done together. So I want to do a couple things within this short presentation is really acknowledge, individuals' efforts that have built PSTC to where we're at. Also relate that to our history and what we've built upon. Talk about accomplishments and impacts, and really, like Amita did, go beyond just these qualifications or regulatory impacts, right? Those are very, very important, but we've done other things that have really pushed the science forward, and we need to highlight those and celebrate those. And then what I'd like to do is really wrap up with two points, right? One, the evolution of our scientific and regulatory strategy, as well as looking towards the future of what PSTC is going to become. So again, same slide that Nick just showed a couple of minutes ago. But what I want to highlight are a couple of very important things, right? Number one, this whole idea about translational safety strategies that accelerate drug development. I think that's a really important statement, right? I think it gets to the root of what we're trying to do as PSTC, right? Sure, we're trying to qualify these novel biomarkers and have them used as drug development tools. But really what we're trying to do, if you think about it, we're trying to change the way we do drug development, right? We're trying to do drug development better. And I think that's the important aspect. Ultimately, by doing drug development, we're going to accelerate the development of safe therapies for patients in need. And that's what it's all about. That is our public health edict that we're following, right? That's what we want to do as the PSTC, plus do a lot of cool science as we work through to this final goal. Again, PSTC brings together pharmaceutical companies to share and validate innovative safety testing methods under the advisement of US FDA, EMA, and PMDA. Exactly. That's what we do. We create a different relationship among our consortia members, a collaborative relationship. We also then can have a different relationship with the health authorities. We can get into discussions that are not directly related to products, but instead related to regulatory pathways that can help then accelerate products, but indirectly, right? Indirectly in many ways, right? GLDH, for example, and it was a perfect example that Nick gave just a few moments ago, is going to benefit drug developers that are working in the muscle disease space. We're not talking about specific the drug being used, but instead creating a better regulatory pathway to go ahead and develop these drugs. So let me jump into acknowledgements and a brief history. So here goes the leadership of c-path over the last 15 years, starting off with Jeff Jacob. Bill Mattis played an important role during the development of the kidney safety biomarkers and the non-clinical qualification. Likewise, Liz Walker did as well. Esley really, expanded the group quite a bit from during her tenure to be able to work on different approaches. Likewise, what also happened was there was an evolution of the qualification process. Liz had to step back in for a little bit, but then I joined PSTC in 2013, where I've now, been lucky enough to spend 8 years of my scientific career. And it has been very, very rewarding. And let's talk a little bit about that reward in a second. But before we do that, we've also had some excellent support scientists and support coordinators across PSTC. We have a very strong staff currently, and we've had very strong individuals that have worked within c-path to help support PSTC's activity across the last 15 years. And a really special thank you to these folks. I mean, these are the folks that make PSTC work, right? From organizing meetings to organizing our science to making sure that we show up on time to our various regulatory interactions. Likewise, a special thank you to all the various consortia that we have collaborated with. And here are a few external consortia. Of course, there's also, internal consortia like the DRISC, the Duchenne's regulatory science consortia that we've been working with around safety biomarkers and hepatic injury biomarkers. But, we've gained a lot from these collaborations. And one of the things that I like to say is, hey, we're c-path, we love to collaborate. And so we want to work with other groups to make them stronger because they also make us stronger. I think there's a couple really nice examples here, right? The Innovative Medicines Initiative Safety collaboration was a great collaboration. I mean, there's no doubt that many of our members were involved in this IMI collaboration, but we as PSTC were also involved. Likewise, our collaboration with the FNIH Biomarkers Consortium has been a very powerful collaboration around the clinical kidney safety biomarkers. A more recent collaboration has been with the Japan NIHS group, where it turns out that we've been doing very similar activities around GLDH, and we were able to synergize our activities, and thus we've developed a data set in Japan as well as in the West that's comparable, which is going to help us then qualify GLDH not only with FDA EMA but also with PMDA. We've done some very strong work with HESI. We haven't done a lot recently, but I think there's always that opportunity to collaborate when we have that opportunity. So also a couple new collaborations on the forefront, collaboration with IMI Transbioline. We're working towards that. Again, a lot of the IMI Transbioline projects are very similar to what we're doing within PSTC, and we need to figure out a way to best synergize those activities. We don't want to repeat each other's work again and again. Instead, we want to figure out how we can best work together in this space and make the most for our effort. Likewise, we've been looking at ways to go ahead and work with the United States Pharmacopoeia around optimizing some of our assays and for biomarker analysis. And one of the examples or one of the pilots that we've pulled together is around the kidney safety biomarkers. We're also beginning to expand our scope a little bit thinking beyond just in vivo biomarkers, but also now thinking about in vitro systems that can help us understand drug-induced toxicities. Again, it's a little bit experimental now, but can we leverage our deep knowledge of these in vivo biomarkers to make these complex systems, more useful for what we're trying to do within drug development? Again, another big thank you to our members, right? Without member participation, we wouldn't be a consortium. The members drive the consortia. This is their consortia. c-path, PSTC staff is here to support the consortia as well as participate in the discussion. And I appreciate everyone's deep participation. The sharing of data is just phenomenal. I mean, I know Amita went through a slide that talked about some of the reluctance in sharing data, and that reluctance is really sharing data publicly. We have amassed substantial data internal to c-path to share amongst the members, and that's a lower hurdle, right? Because the members realize that there is confidentiality around those data and how those data are actually going to be used. Likewise, partners like EMA, US FDA, and PMDA are absolutely invaluable. These partnerships, this collaboration PSTC would not happen without it. And as I said, we can have very, very different conversations with health authorities than individual companies can have. And I know that our members are leveraging that in a good way, right? To make sure that their perspectives are understood. And likewise, the health authorities are doing the exact same thing. PSTC is a vehicle for the health authorities to be able to communicate in their ideas, objectives, viewpoints around different issues that don't have to be specifically compound related and can be in a larger fora such that it doesn't have to be repeated across each individual company. And so these relationships are so important. This collaboration is so important for what we're doing and how we're trying to change the drug development landscape. Here goes just a little factoid for everybody. But, when I joined PSTC, I was looking at our membership agreement. And lo and behold, the membership agreement said the Toxicogenomics Cross-Validation Consortium. Well, it turns out that was the original name for the Predictive Safety Testing Consortia. At some point, it morphed. And actually, if you look at our most current membership agreements, they actually do include the name of the consortia as PSTC. So some things change, right? And some things change for the good. Because I like PSTC better than TCVC. But some things don't change. And I know this is a slide that Nick used, but I'm going to talk about a little bit differently. If you think about c-path and what we do, we're really a one-trick pony. And I don't say it in a bad way, I actually say it in a good way. Because this trick can actually be applied across multiple approaches, right? And that's really our ability to pull in data, standardize that data, and then use that data for some purpose, regulatory endorsement or otherwise. But what I'd really like to concentrate on this slide is what's happening at the very front, right? The consortium study data, non-clinical and clinical. That's the data sharing part, right? These are data that are derived from pharmaceutical company studies that are actually contributed to c-path. Some of this is done prospectively. So studies are run at one consortium member company. Those samples are analyzed for their biomarkers at another one. And then ultimately those data, being the biomarker data and histopathology data, are actually then analyzed at a third data, and correlations are made around whether or not that biomarker is doing what we hope it is doing. Who would imagine such type of collaboration to generate these data sets? Likewise, companies are funding, studies at CROs to go ahead and create this data. So it's just not in-kind contribution through direct work, but also through indirect work, which is highly collaborative, right? In order to drive this biomarker qualification process. Also, at the end, when we get all this data and we organize this data, it's really the member companies working together to come up with those final conclusions and actually putting pen to paper to write those regulatory documents. Luckily, we have good project managers and project coordinators, as well as other scientists to help, organize that. But it's really, at the end of the day, using the knowledge of the various scientists that are participating from industry to drive these documents. So let's shift gears. I'm not going to spend a lot of time on accomplishments because I think Amita did a really good job at that. And what we put together is what you can see here are some infographics, which kind of talk a little bit about, what we're doing and how we're doing it, right? I know I have 14 here. That's an error. That's my fault. We now have 13 member company. But Nick said we have 6 target organs that we're pursuing, which make up 6 working groups then. We have a number of samples that have been shared across the consortia, right? This is important, right? Because these samples can be used and analyzed differently as we go through. But what we also have is all the data from those samples, specific to given organ groups. what this has resulted in is really 15 regulatory biomarker qualifications or letters of support, and well over 40 publications. And so I think what this does is this really shows our accomplishments over the past several years. But let's talk more about that impact piece as we go forward. So when we think about these accomplishments, we can talk about those regulatory endorsements, but then also something that Amita brought up that I think is really, really important is that the impact that PSC, along with others, has had around how the biomarker qualification actually works, right? But the whole process piece, the whole expectation piece. And this is not only across, US FDA, but it's also across EMA and PMDA. We're helping to drive these processes, and more importantly, the scientific thinking behind it. And so we've spent a lot of time looking at what the evidentiary considerations are for biomarker qualification, how we need to interact with the agencies. Likewise, we've spent a lot of time thinking about assay validation and those approaches and expectations. Amita brought up the statistical analysis approach, and that's also evolved very nicely. And there's a recent publication that's going to come out that compares some of the approaches statistically that can be used. But also, I think what we've done too is we've educated stakeholders around, how biomarkers can be used, and shared knowledge around how to basically apply these biomarkers as PSTC. We've also enabled the use of biomarkers. And so here goes the laundry list of various regulatory endorsements. I mean, they're very important, but you can, as Amita said, you can see those on the website, right? And we've had a lot of impact across time, with these various endorsements. Again, we had the very first letters of support, or we had the very first qualification as PSTC from FDA, PMDA, and EMA around the 7 rodent biomarkers for kidney safety. Likewise, we received the very first letter of support from both FDA and EMA, again, around rodent biomarkers. But I think this really changed the paradigm in many ways, right? It gave us another way to highlight the activities that are going on within PSTC before we actually receive a formal qualification around some of these data. Likewise, it gave us an opportunity to, across, the biomarker stakeholder groups, be able to share this information more broadly. PSTC was also, along with collaboration with the FNIH Biomarkers Consortia, received the very first clinical qualification of safety biomarkers, again, around the kidney. And then finally, I'll point out one other significant first, and then that's really receiving a letter of support from EMA around a clinical safety biomarker, and this is for GLDH. And this is a very cool letter of support because basically it acknowledges how to use GLDH as we described in our qualification documents. And so this letter of support, I think, went well beyond any other letter of support in endorsing the use of a given biomarker without actually being a qualification. As Nick alluded to, and Amita, we have a lot of ongoing activities, right, across skeletal muscle, liver, kidney, pancreas. Many of these activities are with FDA, and you can see here with where they're at. But our objective here is not just to have qualification of these biomarkers with a single regulatory group, but instead to have worldwide acceptance of these biomarkers. And so what we're seeing is that as our regulatory activities progress, we're also then engaging EMA, and we try to create a parallel mechanism, a parallel approach between FDA and EMA, and hopefully that's what we're actually going to be able to do also with PMDA. And so you can see that there is overlap between EMA and PMDA, not temporally, but at least across the projects that we're looking at with GLDH and the kidney safety biomarkers. So I think another area of impact, just besides those regulatory outcomes, is really around, as I mentioned, the education and knowledge sharing for the use of biomarkers. So, you know, with the kidney safety biomarkers, we've been able to develop a biomarker user guide, which allows drug development sponsors to go ahead and apply the biomarkers in an appropriate manner. And it really, within that document, that guide, it talks about our learning and experience of how to go ahead and utilize those biomarkers. You know, likewise, we've made, with the letters of support, we've made data publicly available for drug development sponsors to utilize when they're discussing these biomarkers with FDA, right? Likewise, we've had over 40 publications where we've really tried to talk about the secret sauce of biomarker qualification, but then also the basic science behind how those biomarkers are working and what we know about those. As Amita mentioned, we've hosted focused work groups and webinars on the biomarker process, on biomarkers, et cetera. And then also what we've done is we've acted as within, you know, c-path PSTC as scientific consultants for other drug development sponsors to go ahead and implement biomarkers. You know, as far as enabling the use of biomarkers, we've been working with vendors to ensure assay availability. Likewise, we've also been thinking about how these biomarkers can be used outside of drug development and thinking about how we can enable, you know, assays for clinical practice. And we can probably talk a little bit about that within the panel session. Also, we've been working on how to best share our biomarker learnings and know-how, right? And that's that biomarker data repository. I think that is a great mechanism to go ahead and really share those learnings, as well as PSTC in and of itself. You always got to read the fine print, and I think this is an important aspect, and I know that Yuri in his presentation, he's going to go through this. And that, you know, is that, you know, regulatory decision-making biomarkers can be used on a case-by-case basis in IND filings, in IND studies. This is important. It means that you don't have to wait for a biomarker to be used. If indeed the way you want to use it, there's enough data around it to be able to use that biomarker, right? So that's really important. And it's also important that we're sharing all this data, right? So that then enables drug development sponsors to go in and have a conversation with health authorities around applying these biomarkers and using them to make regulatory decision-making. Just an idea there, so always read the fine print. BMDR, I think, is a huge advancement, but yet it's still an untapped resource, right? What we want to do here, as has been explained previously, is really be able to collect novel biomarker data, standardize that data, right? Both preclinical and clinical data, and then share that data across the field in an appropriate way, right? So that we can all learn from those data, right? And so what we're encouraging is to go ahead and have drug development sponsors be able to share their data. It would be, you know, blinded, anonymized, de-identified, of course, and then aggregated. Then these drug development sponsors would then have access to the data and be able to learn from each other's data. You know, as Amita showed, this has been endorsed by FDA in a letter by Janet Woodcock, and really it came out of a conversation, you know, with FDA and IMI and c-path leadership several years ago, right? That this would be a good idea as a next step in how we share data more universally. Again, another impact is really around our activities in the bio and driving forward how the biomarker acceptance process works. And I'm going to save this really for my next section and begin to wrap this talk up. So Several years ago, I remember Janet making this statement and it really rang in my ears. And I think this was really the start of the evolution of what we were trying to do, right? And then what she said was during one of these more difficult conversations around the fact that, you know, we were fumbling around with a number of issues and trying to get things to work. You know, Janet said, hey, look, you know, we're making this up as we go along. And she was absolutely right. We were in a very, very innovative space. But now we have the benefit of 15 years of experience, and we can now begin, and what we've done is take that experience and begin to build better processes and better approaches around the science and around the regulatory acceptance of biomarkers. And I'm going to show you that evolution. This was a really important slide that came out from the biomarker qualification pilot project. It did a couple things in my mind. Number one, it delineates the importance of the context of use, how you're going to use these biomarkers, right? And that also then delineates the amount of evidence that's needed for a given qualification. So I think that that's really important. It also created a division between the two activities that are going on in parallel during, you know, biomarker qualification. That's analytical validation, right? The validation of the measurement technology for the biomarker, and then the clinical validation, demonstrating that the fact that the biomarker actually does what we say it does. And so this was a really important step. I mean, I know now we look at this and go, yeah, sure, that's what we do. But I don't think it was ever delineated before the slide really came out and codified the way it is here. Likewise, another very, very important step that occurred during the evolution of the biomarker qualification processes is the activities that happened during 21st century cures, right? There's no doubt that these activities were happening before the bill was made an act by the U.S. Congress, and that these changes were happening, you know, across FDA, across EMA, and in part PMDA. But what Section 507 really did was it really codified how we can think through a qualification program, right? You know, we knew that we started off with a drug development need. We needed to derive a context of use that would then derive the level of evidence that was required. Ultimately, then we would end up with the qualification of that biomarker, an agreement between the submitter or the requester and the health authority, and then end up with an endorsed tool. That's perfect. But what we also ended up with Section 507 is really a really refined process for a letter of intent to define what we're trying to do, an agreed to plan of how we were going to demonstrate that the biomarker actually performed as we hoped, and then a way to pull that all together into a package for final regulatory review. Now, many of us, and you can see that there are many PSTC as well as c-path authors here, worked very closely with FDA to really refine that process. Right, there was a very important paper that was published in Science and Translational Medicine that defined what came out of a evidentiary consideration workshop, and this was ultimately then moved into a draft guidance document, and that's important, right? To codify how to work through this process, and to also codify what the expectations were to be successful in this process. I'd like to show you something here, and this is kind of fun, and I think it's self-evident once you work your way through it, but I don't think it was self-evident when we started out and looking at how we develop expectations for assay validation of biomarkers. You know, if we look at this a line on the right, I think it really shows what we're thinking about, right? So we have an initial assay that we work out, right? And this assay is imperfect because we don't know a lot about the biomarker that we're actually measuring. But with that assay, we can go ahead and learn about the biomarker. Once we learn something about the biomarker, such as maybe the basal state of that biomarker in normal healthy volunteers, that can then add to our learning and what we need to do to modify that assay. Perhaps the assay wasn't sensitive enough to be able to detect all in all healthy volunteers that basic level. So that assay gets modified. Then we go a step further, right? Maybe we take samples where patients were exposed to a toxicant. And then we go ahead and learn that, wow, these look like they're the high levels that we're going to see following exposure to a toxicant. And here goes a break point, hopefully, between the healthy volunteer reference ranges and now this response from a toxicant. And we can go back and modify the analytical assay. And so by doing this iterative approach, what we're going to end up with is really a fit-for-purpose analytical validation. Likewise, this will then result in a fit-for-purpose clinical validation of the use of that biomarker. And so that's how those two approaches are really intertwined. So if you remember that earlier slide, even though we broke out analytical validation and clinical validation into two groups or two areas, really they're very much entwined. And that's an important learning that took place across time and how we need to intertwine these approaches. Again, you know, this really talks about the life cycle that we go through and how we ultimately end up with that fit for purpose validated asset that we can really begin to look at how this biomarker is performing and how we can then apply that assay to be used in drug development studies. And what we did was we codified this in a points to consider document, a white paper. And if you look at the authors, you know, what you see are authors from PSTC companies. You see from analytical companies, some of which we've collaborated with as PSTC. And then most importantly, you see authors from US FDA. This was also reviewed by EMA and PMDA, as well as other scientists. And so, you know, this was a really important step forward for us in really codifying how we want to do these types of measurements. Again, you know, thinking about clinical validation, you know, there's certain aspects that we need to build into a clinically validated biomarker, right? Most importantly, the fact that it correlates with the truth, that biomarker, right? That hard endpoint. And I think the other important part too is that, you know, not all biomarkers are going to be qualified, even though we love those biomarkers. Some biomarkers just aren't going to work. And we have to realize that, right? There's certain performance characteristics that need to occur or that need to be embedded in that biomarker in order for it to be acceptable. A year ago, we published a really nice paper that really gave away the secret sauce, if you will, of how we do biomarker qualification in the safety biomarker space. And this is around translational biomarkers, how we utilize the non-clinical data to basically then bridge us to our human studies. And I think this is an important paper because it really pulls together a lot of the aspects that we've been talking about for the past several years. You know, likewise, you know, it goes into further detail around how we're looking at histopathological response for a target organ of interest, right? Looking at selectivity, you know, hopefully ending up with biomarkers that are not mechanism of toxicity dependent, how we basically correlate histopathological changes with the response of biomarkers in animals, you know, using histopathology really as the truth and this correlation. Again, you know, one of our favorite approaches to really use the receiver operator characteristic curves to be able to demonstrate the superiority of our novel biomarkers over the current standard biomarkers. And then what we do is we bridge to humans. In humans, we don't have the luxury of using histopathology, right? Like we do in animals. But what we can do is we can use medical adjudication as well as the assessment of the to use drug-induced tissue injury samples, but also disease-induced tissue injury samples. And that's taken us down a really interesting road that I think we can talk about within the panel sessions. So again, the future, and let me wrap up with the future. And it's really going to be talked about, in our webinar series, right? Number one, education, right? How do we actually use the biomarkers that we have that are qualified? Also thinking about how else can we leverage this interaction, this collaboration that PSTC and c-path represents, right? And what are more tissue biomarkers, but then also, in vitro assays, other approaches that we can utilize. Also, and I've alluded to it a little bit in my talk, how can we now leverage our learning about safety biomarkers, in the disease space, right? Can these biomarkers also be used as efficacy biomarkers? Also, how do we accomplish better data sharing, right? That's a main objective of the BMDR, but I think it also needs to be an objective for how we move forward in the future. And then, of course, collaboration begets innovation and additional collaboration. And so we've been lucky within the PSTC where we've been able to spawn additional activities, and we're going to cover those. So I'm going to wrap my talk up there and thank everyone for their attention.